So before we get started, I just want to talk about what iGEM is. So iGEM stands for International Genetically Engineered Machine.
Stand a little closer to the mic. Can't hear it.
Hello? Okay, there we go. Sorry. Okay. iGEM stands for International Genetically Engineered Machine, and it is an annual worldwide synthetic biology competition for undergraduate, graduate, and high school students. And teams basically come together to tackle problems within their community and around the world, and work both inside and outside the lab all summer long to develop these projects. So there are multiple components that go into an iGEM project, from education and public engagement, hardware development, integrated human practices, and developing a variety of measurement and modeling for systems and new parts and software tools. And then, of course, we as team members also gain a lot of different skills, from laboratory to teamwork and team management and problem-based knowledge and so much more, and get to work with fantastic people for really intensely for 4 months straight. So now onto our project for 2018-2019, Honeybees. So our relationship with bees is so long and complicated that it predates agriculture. And yet in recent years, this has all been in jeopardy, and bees have started to die at an alarming rate. So our project this year involved the creation of a detection system, the bee detector. Now, as you know, humans have been keeping bees for thousands of years, and as a result, we have become dependent on them for our survival. Today, bees pollinate a third of our food, feeding 90% of the world's population, and fuel a multi-billion-dollar industry, which has created thousands of jobs globally through their ability to pollinate and produce honey and wax. But in recent years, the survival of the honeybee has become a source of international concern, as the honeybee, along with all other bee species, are currently under threat and dying off at record rates. In 2018 alone, beekeepers in North America lost an average of 40% of their colonies. And to put that in perspective, a loss of 18% is the maximum loss that a beekeeper can experience without the loss being considered abnormal. And as scientists have warned us, the loss of pollinators is serious, as it could lead to the collapse of entire ecosystems, threatening our food security, jobs, and harming us both socially and culturally as well. Now, the question at this point is, what is killing the honeybees? And through extensive consultation with beekeepers, we learned that there are numerous issues affecting them, but particularly concerning is Nosema ceranae. Nosema ceranae is an intracellular parasite that infects the midgut of the honeybee, and the way it works is once the honeybee ingests the spore travels to the midgut, where the infection spreads to the rest of the body, wreaking havoc on all the bee's systems. This causes a multitude of negative effects which can be lethal, including an alteration to the bee's metabolism, energetic stress, bodily malformations, and a loss of cognitive function. And if all of these don't kill the bee directly, then Nosema makes the bees more vulnerable to other stressors, such as extreme temperatures, lack of food, and other diseases. Meaning that Nosema is particularly concerning, as it can worsen all the other factors affecting the hive, particularly in colder temperatures like those for us in Alberta. These effects also have massive consequences on the hive as a whole. It's like a positive feedback loop that isn't so positive. As more bees die, the colony is weakened, which then makes uninfected bees more susceptible to infection, thus transmitting the debilitating effects to the entire hive and lead to the collapse of entire colonies, making Nosema ceranae a leading cause of death for the honeybee. In 2018, our team developed our project APIS, our antifungal porphyrin-based intervention system, a treatment for Nosema ceranae. But after we finished our project, we realized a detection system was lacking, which is why we looked into creating one. We grew particularly close with the Alberta Beekeeping Commission, and after presenting at your conference last year, we realized that while APIS helps with battling Nosema ceranae, we should continue on the issue of Nosema to tackle the issue of detection, as there was no effective way to detect Nosema. And as Alberta is the largest honey producer in Canada, this became a huge motivating factor for us to continue and help the beekeeping community as much as possible. But of course, when deciding on our project and designing our detection system, we wanted to consult with our stakeholders from across Canada. And it was by speaking with over 9 beekeepers, including Tim, Barry, and Stefan, that we realized detecting Nosema is an issue that matters to all beekeepers. Because if you can't detect something, how can you treat it? Currently, there are 2 common ways to detect Nosema. The first is through visual signals. This is the only on-site detection method and requires beekeepers to watch for declining populations, poor honey production, dysentery in and around the hive, and worker bees crawling around the hive with swollen and greasy-looking abdomens. And I can imagine that it's not particularly easy to find to figure out if a few of your bees are greasy or not one day. And unsurprisingly, every beekeeper we spoke to told us that this visual method of detection is inadequate, as often these symptoms only appear once the hive has reached a point of infection that can no longer be treated. And this was further confirmed by Courtney McInnes, a bee researcher that we spoke to, and has helped us immensely with our project. And as Tim, a hobbyist beekeeper, told us, hobbyist beekeepers are sometimes unaware of the symptoms and as a result don't watch for them. He, along with many other beekeepers, said that this was scary because it often leads to people who are trying to do a good thing for the bees, causing more harm than good as the infection gets out of control and are spread to nearby hives. And as Barry, a commercial beekeeper, told us, this worries commercial beekeepers as they can lose upwards of 40% of their hives to Nosema during a bad season. Now, for wealthy and experienced beekeeper with time to wait for results, the second detection method, which is also the gold standard, unfortunately, is sending off a sample for microscopy at a government or commercial lab. But beekeepers also find this method inadequate as it is time-consuming, costly, and as Stefan told us, it can take upwards of a month to receive results. So, in response to the massive problem of detecting Nosema ceranae, we created the BeeTector, our accessible, easy-to-use, and on-site detection method for Nosema ceranae. We wanted the BeeTector to be something that beekeepers can use in the field. Through many consultations with beekeepers themselves, we learned that they were very familiar with and would prefer using paper strip detection methods, like pH strips, which they regularly use for testing their nectar and honey. We also learned that they're comfortable with crushing up a handful of bees without worrying about their numbers, as at least 2,000 new bees are made in a hive each day, and it's something that is required if you were to send your samples for lab testing as well. What we wanted then is basically something like a pH test, where you dip a paper strip into a sample and then look for a signal to show that you have a Nosema infection. Like a pH test, we designed the BeeTector to be simple, familiar, timely, and field ready. But how do we detect the Nosema spores? We found that the surface of the spore is poorly characterized, which posed a challenge for our detection system. We faced this challenge by finding a ligand to the Nosema spore using Phage Display, which has never been done before. So what is Phage Display, you may ask? So Phage Display is a laboratory technique for the study of protein-protein, protein-peptide, and protein-DNA interactions that uses bacteriophages, which are viruses that infect bacteria, to connect proteins with the genetic information that encodes them. And this is a technique that is commonly used in immunotherapy. We then aim to tag the protein with a reporter. We aim to tag the phage with a reporter protein, giving a visual signal on our paper device once they bind to the spores. Now, to achieve these things, we further divide our project into 3 main components, with these being the spore ligand, which will provide specificity for Nosema spores, the reporter phage, which provides quantifiable visual signal, and the paper strip, a familiar implementation method. The spore ligand component involves the ligand expressed on the surface of M13 phages that bind onto Nosema spores. To select for potential candidates that can bind to the spores, we performed a series of biopanning experiments using a 12-mer peptide commercial Phage Display library. The power of this technique is that you can test many M13 phages expressing 1 billion unique peptide sequences that are 12 amino acids long at the same time. We selected potential binder candidates by mixing the phage library with Nosema spores, and the non-bound phages are then washed away multiple times, leaving the phages bound to the spore. Those are then, with an acid elution, we then separate the phages that are bound and use them as the input for the next round of biopanning. And in this way, with each round, we are able to enrich for better spore binders, resulting in higher specificity. So each spot that you see on the plate is a different phage acid eluted from spores in our third round of biopanning, displaying a promising candidate ligand for the B-tector. To make sure that our phages did not bind onto uninfected bees, giving a false positive result, we performed a negative selection experiment with a known uninfected bee sample and tested the sample with our stronger candidates from the third round. This experiment found no binding of any phages to a known uninfected bee, thus showing specificity to the peptide to the Nosema spores. The next component was the reporter phage, and which involves tagging the phage with a chromoprotein to give off a visual signal for the spore binders. Expressing such a big chromoprotein like Imagelyme onto M13 hinders the assembly of the phage. Due to this, the component itself is further split into 3 parts. The phage spore binders tagged with one peptide, the Imagelyme chromoprotein tagged with another peptide, and the sortase enzyme, which binds these tags together and conjoins Imagelyme onto the spore binder. We further characterize our chosen chromoprotein Imagelyme for its fluorescent properties, which also helps us quantify the B-tector during the experimental process and production. So the Imagelyme characterization was performed for its quantum yield, emission, and excitation properties, as seen on this graph. For the visual signal resolution of the chromoprotein, we also measured the fluorescent properties of serial dilutions of the chromoprotein to model the Imagelyme quantity in the B-tector that produces a visual signal. The final component of the B-tector system is the paper strip, which allows the separation of phages bound to Nosema spores from the unbound ones, as these can still produce a signal as they still have the chromoprotein. So if there is no infection, the phages have nothing to bind to and would elute all the way to the top of the paper. If there are spores in the bee sample, the phages bound to it would move less through the paper and appear as a band at the bottom, whereas the unbound phages would migrate further in the paper to the top. And in the case of a strong Nosema infection, a more intense band would be visible at the bottom and no unbound phages at the top. In the lab, we tested different types of paper and selected one that could visibly separate the components of a crushed bee sample. Moreover, we tested mobile phases of different polarities, noting that the chromoprotein alone can migrate through the paper different with those. Overall, these results made us confident our separation strategy works for the B-tector application. So as a whole, the beekeeper collects a sample of bees from a hive, crushes them, and then mixes them with our B-tector solution. Then the beekeeper dips a paper strip into that solution to separate the components of the mixture. A positive sample for spores shows color at different intensities and regions of the paper compared to a negative sample. The intensity of the color would directly correspond with the severity of the Nosema infection. Super simple, right? However, coming up with this system in a lab environment is not indicative of how it will work in the field, which is why, to address this, we developed a prototype and designed a detailed protocol that any beekeeper can essentially use. A central part of our project design, after all, focused on the end user, the beekeepers. And as our diagnostic system had to be simple, familiar, and timely for out in the field. So when we got in contact with Paul Christensen, a commercial beekeeper, to test our prototype in front of us, the prototype testing was done at their house, a setting that would be comfortable and is similar to where our final product would be used in real life. And through watching Paul use our prototype, we were able to use his very helpful insights and inputs to improve our final product design that we had not even considered before. And one of the questions that was raised was how many bees are actually needed in order to produce a signal. And looking more into this, our team created a model to answer this question. So we chose a signal analysis model, and based on our conversations with beekeepers, we acknowledged that all of them advocated for a time-efficient on-site test to identify Nosema spore count in a simple manner. And by characterizing our signal, we want to establish how strong our ligands are, the detection limit of our system, and the correlation with the level of infection. With this, we can provide the beekeeper with specific instructions on what they should be looking for when using the B-tector, making our product as ergonomic as possible. By obtaining adequate results from the biopanning experiments, we can provide a spectrum of visual signals indicating different spore counts. The beekeeper can then compare the paper strip with this spectrum to determine the level of infection. We know the minimum amount of spores that correspond to a diseased bee. We wanted to investigate if our system can visually respond to that. Our variables are the number of chromoproteins per phage and the number of phages per spore. For the number of chromoproteins per phage, we are using literature values. As per the number of phages binding to a spore, we are solving for a range. From the minimum of 1 phage per spore to the maximum, the optimal number of phages that can spatially fit on a spore. This was determined after a series of computations where we considered factors such as surface area of the spore, phage dimensions, and volume of space available to the phage for binding. Our expected value would be anywhere in between. The target is to be able to visually differentiate the Imogeline band containing unbound phages from the Imogeline spore band containing the bound phages. Based on results of the biopenning experiments, we are inputting our values here to correlate it with our signal, and then calculate the sensitivity of our detector. This can help us evaluate if our ligand is adequate for this specific application. It was determined from our teeter assay that an acceptable ligand must provide a ratio greater than 15 phages per spore to visually differentiate between bands. Moreover, the ideal ligand for our application is the one that provides a ratio of phages per spore closest to the optimal packing case. So to answer Paul's question, how many bees are needed, our numbers are considerably high. To address this issue, we came up with potential modifications that can improve the performance of our product, such as a reporter protein with smaller in size with higher visibility, and or a phage with the highest binding affinity would improve the quality of the visual signal. Furthermore, the volume of detector solution can be decreased by 100-fold to reduce the bee input needed to detect an infection. Now, aside from our signal model, we also wanted to investigate the most efficient method for the production of the detector. And so we modeled the kinetics of this model to compare different production scenarios. This enzymatic process would be composed of 3 reactions, which characterize the binding of proteins and enzyme recycling. So a simultaneous process and a sequential process were investigated, and they both seemed to work. However, according to the simulations we performed, a faster production rate was observed when all 3 reactions happened sequentially, in contrast with the simultaneous process. As there's a lot of room for improvement with this model, we envision to enhance its usefulness by including applicable variables such as diffusion times and control temperatures. So now, we have to ask ourselves, is this detector really that safe to use? Well, to reassure the safeness of our product, our system will provide a quenching solution with specific conditions to kill every single phage on the used paper strip. The beekeeper can then dip the used paper strip full of phages onto the quenching solution to dispose of it in a safe manner. Part of our project involved community engagement, and we spoke to schools and summer camps across Alberta about our project, as well as the farmers market. And one of the common concerns from speaking at all of these different places was GMOs, and more importantly, GMO regulations in Canada. So in response to this, our team created a policy review of GMO regulations in Canada. The policy review detailed the areas where regulations could be improved and how to improve them without limiting scientific development. For example, we found that the government has not updated its GMO regulations since 2003, and there is no mention of synthetic biology. From all of this, our conclusion was that when the public raises concerns about GMOs in Canada, they are sometimes legitimate, and as the government of Canada has a long way to go when it comes to improving our GMO regulations. We also wanted to have a positive impact on the scientific community by helping to make STEM a more inclusive place, and we were also able to share our project with all of those who were in attendance. To achieve this goal, we hosted a Girls in STEM night, which featured many keynote speakers, including our first female dean of science, and more than 30 girls to speak in a roundtable setting about the barriers that women, girls, and other gender minorities face in STEM fields and how we can break these barriers. So in summary, the University of Alberta team is working on a reliable, on-site, cost-effective, and accessible test for Nosema ceranae while engaging with and educating the public and improving the iGEM community. Together, the bee detector and APIS can act as a complete system against Nosema ceranae, solving a massive issue affecting our local and international beekeeping community and hopefully saving bees. Please check us out on our social media. And finally, a big thank you to the ABC for allowing us students to pursue a project that is about Nosema ceranae and honeybee health. Thank you.